The overall goal of this procedure is to use artificial antigen-presenting cells, or aAPC, for ex vivo expansion of human antigen-specific CTL for potential use in adoptive immunotherapy. This is accomplished by first conjugating HLA-Ig and anti-human CD28 monoclonal antibody to magnetic dynabeads to make aAPC. The second step of the procedure is to perform quality control and peptide loading of the aAPC. The third step of the procedure is to isolate human peripheral blood CD8-positive T cells. The final step of the procedure is to incubate CD8-positive T cells with aAPC for a week. The CTL are then harvested and characterized before they are either used or are re-stimulated for another week to achieve the desired level of their specificity and expansion. Ultimately, tetramer staining results can be obtained that show that CTL that are expanded in the presence of aAPC have an increased antigen specificity.
Today, I'm going to show you an HLA-Ig-based aAPC system and how to use this system to efficiently expand CMV-specific human CTL ex vivo. Compared with other existing methods, our aAPC technology offers several important advantages. It's off-the-shelf available, it's of unlimited quantity, and the same aAPC can be used for all HLA-A2 positive donors. Okay, let's start.
Transfer 1 milliliter of M450 epoxy beads from a stock, approximately 400 million beads, into a sterile screw-top glass vial. Wash the beads once by adding 1 milliliter of borate buffer and then placing the glass vial against a dinyl magnet MPC1. While the beads are adhered to the side of the vial, remove the supernatant by aspiration. Resuspend the beads in a mixture of borate buffer, HLA-A2-Ig dimer, and anti-human CD28 monoclonal antibody. Then, to facilitate protein-to-bead conjugation, rotate the glass vial on a rotator at 4 degrees Celsius for 24 hours. Next, place the tube in an MPC1 magnet and remove all of the borate buffer. After the buffer has been removed, wash the beads twice with 1 milliliter of bead wash buffer. Now, incubate the beads in 1 milliliter of bead wash buffer and rotate the vial at 4 degrees Celsius for another 24 hours to block the residual protein binding sites on the beads. Then remove the buffer from the glass vial and replace it with 1 milliliter of fresh bead wash buffer. Transfer 100 microliters of FAX washing buffer to FAX tubes and then add 5 × 10⁵ beads. Stain the beads with 1 microliter of anti-mouse IgG1PE and 1 microliter of anti-mouse IgG2A FITC. After staining for 20 minutes at 4 degrees Celsius, add 3 milliliters of FAX washing buffer to each of the staining samples. Then pellet the aAPC by spinning at 300 Gs for 5 minutes at 4 degrees Celsius. Resuspend the aAPC in fresh FAX buffer and read the staining result immediately by flow cytometer. Wash the beads twice with sterile PBS as before. Next, load the beads with 10 microliters of CMV peptide. Count the beads by hemocytometer and then label them with the date and concentration of the beads as beads per milliliter. Incubate the aAPC with the peptide for at least 3 days at 4 degrees Celsius. Centrifuge approximately 100 milliliters of fresh peripheral blood from an HLA-A2 positive donor in 10 sodium heparin vacutainer tubes at 300 Gs for 10 minutes at room temperature. Carefully remove the top plasma layer by aspiration. Replace the collected plasma with sterile PBS and transfer the blood into a sterile T75 culture flask. Mix the blood and PBS well by pipetting. Once all the blood has been collected, add 15 milliliters of Ficol Pac Plus to 4 50 milliliter conical tubes. Slowly overlay 30 to 35 milliliters of the blood cells on top of the Ficol in each tube, keeping the interface distinct between the Ficol and the blood cells. Centrifuge the blood and Ficol gradient at 500 Gs for 20 minutes at room temperature, with the brake off and the acceleration as low as possible to maintain a clear interface between the layers. After the spin, aspirate off the top plasma layer and then use a serological pipette to carefully aspirate the PBMC layer. Transfer the PBMC to a fresh 50 milliliter conical tube. When all the PBMC have been harvested, add 30 milliliters of PBS to the cells and centrifuge them. Next, discard the supernatant and wash the pellet one more time with 30 milliliters of PBS to remove the residual Ficol. Then enrich for the CD8-positive T-cell population using a Milteney Human CD8-positive T-cell isolation kit according to the manufacturer's protocol. Count the CD8-positive T-cells and confirm the purity by FACS analysis. Suspend 1 million CTL in 8 milliliters of T-cell growth factor 2x culture medium plus 8 milliliters of complete RPMI medium and add 1 × 10⁶ aAPC. Mix well. Then, use a multi-channel pipette to plate 160 microliters of cells per well onto a 96-well U-bottom tissue culture plate. Incubate the cells at 37 degrees Celsius in 5% carbon dioxide for 7 days. Feed the cells on day 4 with 80 microliters per well of T-cell growth factor 2x culture medium. Harvest the cells on day 7 and then place the cells against the magnet to remove the old aAPC. When all the beads adhere to the wall of the vial, harvest and transfer the cells into another 50 milliliter conical tube and count the cell number. Determine the antigen specificity of the aAPC by tetramer staining according to the manufacturer's protocol. Replate the harvested cells with new aAPC under the same conditions to generate increased cell numbers and antigen specificity. Here is a representative tetramer staining result of CMV-specific CTL generated by aAPC after 1 week of culture. Shown here is a representative intracellular cytokine staining result of CMV-specific CTL generated by aAPC. CMV specificity was 61% by tetramer staining.
This method can be used to answer key questions in the T-cell field, such as how can we identify the optimal culture conditions and requirements for T-cell functional modulation. Though this method provided insights into treating viral diseases, it can also be applied into other fields, such as cancer.